With what we have learned earlier, we can now construct some of the important nonlinear spectroscopic methods. We start with the one which is the most straightforward and also probably the most important of all those spectroscopies. I will not follow in any way how these methods have developed historically. I mean, it's clear when you learn about pump-probe spectroscopy that this is some sort of a generalized absorption, and that the generalization is straightforward even without the theory that we have created. But having the theory and knowing about the problems of heterodyne or homodyne detection of weak nonlinear signals, we can use that motivation to construct pump-probe spectroscopy somewhat differently than it's usual. So our typical idea of a nonlinear third-order spectroscopy at the moment should be the following. We should imagine 3 pulses coming from 3 directions into a sample with molecules. This sample can be whatever. Here are the molecules, and by somehow combining the directions that we have here, we would get a signal that would travel in a direction different than all 3 of the initial directions. So here we would have the Ks, which would be minus K1, for instance, plus K2 plus K3. This is one of the directions into which we can go. Now you can imagine that here is a problem. The signal goes into a direction that is background free, and therefore it will produce this very weak signal that it will be quite hard for us to resolve it spectroscopically. We would like to use the so-called local oscillator field. So this would be the direction of local oscillator, which would be the same as the signal, and mix it with the signal. In this situation, we have a problem, however, namely we don't know what's the proper phase relation between the independent local oscillator pulse and the signal pulse. And we have seen that if we measure the signal with this spectral uncertainty, we get a huge distortion of the line shapes. The line shapes that were purely absorptive, looked basically like absorption, they would now have a component that has a knot. So in the middle of that component there is a 0, so precisely at the excitation wavelength. So we can, however, use basically the same setup, but we can manipulate with the pulses. Basically we see now that the signal is different from the direction of K3 of the last pulse. The reason why this is different is basically that minus K1 plus K2 gives a non-zero contribution. So if we go and basically put together the K1 and K2 contributions into one pulse, for instance like this, so this would be now K1 equals K2, and then we send the K3, the signal will go along the K3, so this is the Ks now, which is equal to K3, and the small signal that we produce, the Es, will mix together with the third pulse. We can actually denote this by PR. It's a probe pulse. So the total signal or total field that we see behind the sample will be K3, that is the K probe, or E3 E probe plus Es, the small signal, and we can now use everything that we have learned before about the heterodyne detection to see what kind of signal we have actually detected. And interestingly, we will see that this is some sort of a generalized absorption and that it doesn't have any phase problem. Yet we are detecting the signal, in fact, by heterodyne detection, that is by mixing it with some strong field. All right, one complication that comes out here is that the present signal field, so let's see, signal field, which is of the third order, is well, it contains not only the third order response, but obviously also the first order response to the pulse K3. So here we don't need to discuss in detail all the prefactors, so we'll just remember that the main dependence of the signal fields on the polarization was this factor I omega, and then we have to expect here first order polarization coming from the K3 alone, plus the third order polarization, which we now know that also goes in the same direction as the P1 and therefore the signal and K3, but is of third order because it is influenced also by the other two pulses. Those two pulses, however, now do not change the direction because they go into the same direction, so K1 and K2, they exactly cancel. Okay, so we have to also add the path that this signal spans in the sample. That's one of the results we have got earlier. We also know that the linear contribution, that is the absorption, can really be expressed in terms of a heterodyne detection of the field that it gets absorbed, which is now the one with K3, and the newly created first order field. So that's the first term that we have. Okay, if we look into the omega domain or the frequency domain of this problem, we can write all this stuff using Fourier transforms. So we have the third order and first order contributions here, and certainly we can write the first order using susceptibility. So we write epsilon 0 chi 1 omega E omega, and that's the probe field here, plus we can write again basically in the same way epsilon 0 and third order polarization, which depends on omega, but also parametrically depends on the delay between the second and third pulse and the first and second pulse. So that's something that we also know how to evaluate, and here we have the probe field. The chi3 here does not depend, I mean, this is not a proper third order susceptibility, because a third order susceptibility would not depend on any field and it would be followed by three fields, whereas here we have some sort of susceptibility of a system which is perturbed by first the K1 and K2 fields. Basically the first pulse that comes in perturbs the system, and we look at the linear response of this perturbed system, and we at the moment do not look at all at how this perturbation by the first pulses look like, except that we know that obviously there will be those parameters that separate the pulses, that they will be involved. Okay, now as with the normal absorption, we can now construct some delta I and divide it by the initial intensities. So it's going to be the intensity that comes out from the signal divided, or from the sample divided by the initial intensity, and we know also, or we will set basically the I0 proportional to the probe pulse square. We will look at everything in the frequency domain. Now this is, let's say, the situation which we would have in the linear absorption. However, and that's quite important, we do not now consider the standard first order response. I mean, when we had the first order response, there was no reference which we could subtract from the problem. But now in pump probe, we have a reference. We can have a look at the absorption that is measured when the system is not perturbed. And that means that, you know, if we take that as a reference, the field that we have to define as I0, that is the reference one, is the field in which the absorption is already accounted for. So we will change the definition of I0, because this is, let's say, when pump, the first pulse, pump is 0. And we will rather use as a reference the situation in which we have probe pulse present plus the first order response to the probe pulse. So this is it. And that's when the pump is 0. But we will assume that the absorption that is present when the pump is 0, even the absorption will be accounted for. Yeah? So we define the I0 as the original probe plus the first order field that comes out of that. That will help us a lot, because then we can go and define the I, the intensity which we subtract this I0 from, as basically, again, this is not equal, but this is proportional to the E probe field that goes through plus the E1 field, which is also included in the reference, plus the E3 field, the third order response. And this depends on EPR. So, to be clear, the original I0, the one that is defined here as yellow, that's a situation when pump would be 0. And in that situation, if we send the probe, we will just measure normal absorption. But because we want to subtract this normal absorption, we put the first order contribution here to the reference. So all this is basically a situation where probe is 0. And when finally probe is not 0, the I would, the signal that comes out would look like the last line, and we have to subtract I0, the newly defined I0 from that, and everything has to be divided by I0. So using these definitions, we can write out explicitly what the I is. It can be clearly written as the reference, which is the probe plus first order plus the third order squared, which we will ignore, plus, because it's too small, plus 2 real parts of the E probe omega plus E1 omega, all this complex conjugated, multiplied by E3 omega. Okay, this is therefore the I0 plus a term which is very small, that's this one, plus 2 real parts of the E probe omega E3 omega. And again, we can assume the combination term of the E3 and E1 as to be very small. So plus, again, very small contribution. So this one term, this is the term that we are interested in, and that's the only term which will basically survive when we subtract I0, and we will now have to rewrite it in some good way in order to be able to divide the I0 intensity away. The delta I divided by I0 can now be written as 2 real parts E probe omega E3, and here we will have to divide it by I0, which for this purpose we can in principle replace by E probe square. So that's what we get for the delta I divided by I0. So here all the prefactors that we would have for the relation between E square and I, they go away because we divide the electric field out. Now we use the definition of the third order electric field in terms of the chi3, that is the third order nonlinear susceptibility of that kind that we introduced, and we get the following. We have two real parts. Here is the probe, and we have epsilon 0 chi 3 of omega and E probe omega divided by, and of course because this is the P, we also have I omega as a prefactor to this. So, and this is divided by E PR square. Okay, and now I see quite clearly that there cannot be a problem with phase in this measurement, because these two terms together, they also give E probe absolute value squared, and that can be easily divided here and doesn't depend on the phase. So now we just have to write out how is it with this I and the real part. We can easily check that if we have I and a complex number and take a real part out of it, that we are getting minus the imaginary part. So for our situation, this will mean that the delta I divided by I0 is minus omega epsilon 0 imaginary part of chi 3 omega. We can write here the parameters. It also depends on, we forgot here to mention that the electric field also depends, the signal electric field also depends on the distance traveled. So there would be this, and the relation between polarization and electric field we have also stripped here of some prefactors. So we would have to write proportional, or we could just invent a constant kappa that will be put in front of everything to just justify that proportionality or express that proportionality. Now, we also know from earlier discussions that delta I divided by I0 divided by the length of the interval, that this is minus the absorption. This would be in the linear case. So by construction that we have made here, whatever we have obtained from the third order susceptibility here would be some sort of a third order absorption, a differential absorption. In fact, it's an absorption that we obtain from the combined action of the pump and probe pulse when we subtract the action of the probe pulse alone. That's the meaning of actually taking as a reference the E1 that was produced by the probe. So putting all this together, we can write for the third order absorption that it is proportional to omega imaginary part of the third order nonlinear susceptibility. And I stress that susceptibility defined this way is actually of the first, it's a susceptibility of the first order in the probe pulse, but calculated or defined for a system that is already perturbed by the pump. All right, so we can of course write all this graphically. We can discuss what's actually happening in the measurement that we have just constructed, and we should also remind ourselves why we constructed this measurement this way. We wanted to get a third order nonlinear signal, which we got, and we found that if we do it in this very general way with 3 pulses going 3 different directions and getting the signal background free, in order to nicely detect it in a way that is similar to detecting absorption, we would have to mix it with the so-called local oscillator field, but we would have a big problem of setting the phase, the correct phase of the local oscillator. And this can be solved by providing for this signal field some sort of an automatic local oscillator, which is the field that generates the third order, the last of the three fields that generate the nonlinear response itself. Okay, so what does that all mean? So in fact, in pump probe experiment, we first measure absorption without probe. So there would be a reference absorption. I'll draw an absorption of some two-level system. And this would be measured in absence of the probe pulse. So this is a reference field. This is absorption in absence of the pump pulse. So this actually measures the effect of the E1 in our discussion. The E1 is the linear response of the system to the probe pulse. A second measurement that we'll do is a measurement of, again, the absorption of the probe pulse, but now after there was an action of a pump pulse. And what can happen is, for instance, in this peak we will have less signal, less absorption, and for instance somewhere here we would have more absorption than we had before. And of course we can take a difference of that because we want to work, we want to get rid of the reference absorption and think only in terms of the third order signal, in which case we'll get the differential absorption, which will be the absorption with pump minus the absorption without pump. So the delta alpha, the differential absorption, will be defined as the absorption with pump, that is the alpha 1, that's the linear absorption of the probe, plus the third order absorption, which depends on the probe and the pump. So the first one is just probe, the second one is pump and probe. And from this we subtract the situation in which we have no pump. So there is basically only the alpha 1 omega. That's the white curve in the picture above. And if we denote this by green, we can see that on one of those peaks we have less absorption, and on the other peak we have initially on one side also less, and then we have more. So we will get some sort of a curve like this for the pump probe spectrum, and we have regions with less absorption and regions with more absorption. With respect to the standard linear absorption. And we will of course have to discuss at some point what does all this mean for the interpretation. So what's the interpretation of having less absorption, and what's the interpretation of having more absorption? So we have discussed differential absorption or pump probe spectroscopy from the perspective of susceptibility. We defined something which looked like a non-linear susceptibility. However, the details of it were a little bit different from the real susceptibility, because the susceptibility of the third order would have to be multiplied by 3 electric fields. But the version that we had was basically a linear response of an already perturbed system to a single field. Now let's write the third order signal field that occurs in pump probe in a little bit more detail. So we first discuss Fourier transform of the signal field, which should be basically the I omega, and again this is probably not really equal, but proportional to, we have some prefactors of with that too, p omega, which is the third order polarization, now Fourier transformed, and we have the optical path through the sample. Now if we write this with our theoretical knowledge about how the p of the third order looks like, it would look the following way. So there is going to be a factor I divided by h bar to the third power. That's from the 3 occurrences of the commutators. And now the Fourier transform of the response. We will write the response with the tilde, that is, we write out here its slowly evolving part, and it will depend on t and possibly also on the capital T and tau. That is basically assuming an ultra fast excitation. So we have time axis and we have pulses that are of delta function type, and they are delayed by t and tau times. So here is time 0, that's where the third pulse sits, etc. So under these conditions we can write here some sort of an R, and again we can, for instance, try to discuss all these things with an example of one response function. So we write one specific response function or Liouville pathway R2 here. So when we are taking an impulsive response, that is the response where the pulses are extremely short, so all the integrations over the pulses will go away and we will only get their amplitudes. And we have two pump pulses, or the second power of the amplitude of the pump pulses, and we have a probe amplitude present here. And then, because R was written with tilde, that is, it's the slowly evolving part of the response function, we also have to add the phase factor that we have already identified to be e to the power of minus I omega eg t minus tau for the rephasing pathway R2. And because this is Fourier transform, there's going to be e to the power of I omega t with that. We will not make all these things explicitly. I mean, no integration will be done explicitly here. Capital T and tau, they are just parameters, so there's no problem. And otherwise, we can see that everything else is exponentials. So we will write this using the R2 Fourier transformed. That's what we mark by here, the omega dependence. Okay, and this whole thing will be basically nothing but the non-linear variant of the susceptibility. Actually, to be completely clear about what's going on here, we should actually keep, or we should be a little bit more precise. There will be omega minus omega eg here, and then t and tau. Okay, so because we have the analogy, so linear absorption analogy, that absorption is proportional to omega and imaginary part of the susceptibility, we would have something very similar here, where the response function is not originating from the linear action but from 3 pulses. We actually know that this term omega imaginary part chi omega, that it corresponds to the minus real part of the field that induces the polarization, that is some sort of a E0 of omega imaginary part, and here the I omega p, and divided by E0 square, and this equals to imaginary part of E star E, E0 star E omega chi divided by E0 square, so it's precisely what we expect. It's the omega and the imaginary part of chi. That's the construction by which we get the linear absorption. So now the what corresponds in here to the third order response function is the Fourier transform, and this will be t and tau, is the Fourier transform of the response function to the third power r2 t and all the other things here. So I'm not going to repeat it, but it's basically all these factors. Okay, that means, or this we can evaluate as minus I divided by h bar to the third power r2, now a function of omega minus omega eg, and also a function of t and tau. And comparing it to what we have above, we can see that we need to take an imaginary part of this to get the third order absorption. So third order absorption will be the imaginary part of chi 3, which is, one can actually see that this is the imaginary part of a function which looks like minus I and then some real and imaginary part, and that this here gives us that we get the minus real part of it. So alpha 3 omega from here and from here will be then proportional to minus omega, that this omega comes from here, minus omega divided by h bar to the third power, and real part, that's the meaning of I here, real part of r2 omega minus omega eg as a function of t and tau parameters. So as a conclusion, we basically see that the contribution from R2 to the absorption gets an overall minus, and it also has to contain, actually all these things are proportionalities, so we also have to include how the action of the pump pulse appears in the response. So here we would have to have a pump to the second power, and here also a pump to the second power, and basically here this is the way how we got rid of the probe amplitude. So here we should also have a pump square present. Okay, so to summarize, the third order absorption as a function of frequency, possibly as a function of t and tau, will be proportional to, and now let's write out also the R2 in terms of the elements of the evolution superoperators. So we will have minus omega a pump square divided by h bar to the third power, and now real part of u, and now we write it of course also only for two-level system, we didn't discuss anything larger than that, eg of omega minus omega eg, and then we had u e of t and u ge of tau. Now what is this first thing? This, I mean, I have used here the notation that if I have something like u, e.g., for instance, that depends on time, and I make a Fourier transform of it, this will be the same function with omega. However, we have already defined this function, or called this function something, and that was the GEG, omega minus omega EG, and that was the line shape. That was the function that defined line shape. Real part of GE, we are actually having a real part here, would give us absorptive line shape for like really a line shape of the absorption. Well, in principle, we can also set t equal to 0 and tau equal to 0, and that would give us, that would basically tell us that the differential absorption is perfectly proportional to the absorption spectrum of the system, because at t equals 0 and tau equals 0, the u's, UEE, EE and UGE, GE, they are unities. Also, we can say that at tau equals 0, there is no difference between rephasing and non-rephasing signals. There's no difference between those two signals, so we can actually sum all the pathways, because if you remember the structure of the pathways, they look essentially always the same in T and capital T, and in tau they have complex conjugation, but as they at tau equals 0 they don't differ. We can in principle say that the third order absorption spectrum, that is the differential absorption, would have the following form. Basically, we will define this absorption so that we will also divide by the pump pulse. So I will add here this additional bar, which will denote this division of the power of the pump pulse, and then we have this quantity proportional to minus omega h to the third real part of the Fourier transformed contributions of the all four positive Liouville pathways. So there will be G, EG, EG, omega minus omega EG here. If we assume capital T and tau equal to 0, then all other factors are 1, and there is no difference between the pathways. So the GEG here represents all four pathways. So this represents R2 plus R3, the rephasing pathways, plus R4 plus R1, the non-rephasing pathways. So in a special case that the capital T is equal 0, this corresponds to a real pump probe situation. I mean, just to remind you of the geometry, we have a molecular sample. We send now two pulses, let's say from here, one direction, there's K1 equal K2. So it's actually a single pulse that we count twice. Actually, we count this twice. This pulse comes first, and second with a delay capital T, which actually we have set to be just 0. We send a second pulse which looks exactly the same, except that it comes from a different direction, from a direction K3, and this produces a signal which we identify, or which we actually isolate from the background by subtracting the absorption that occurs when no first pulse is set in. So here we will measure the delta A, the absorption, which is our alpha 3 of omega. So we have a relatively simple theory now, which tells us that we have to sum up all the pathways and take their line shape. In the simple theory that we have produced so far, they all have exactly the same contribution. The contribution, that's the contribution in time t. The contribution in the time capital T will be different for these three pathways, and we will discuss how later. So far, we set, and that's quite important, we set tau equal 0 because this first pulse actually counts for two pulses, k1 and k2 in our theory. Now, in order to understand what does it mean that we get a minus prefactor here, we have something that evaluates into absorptive line shape, so it's positive everywhere in omega, and it is multiplied by minus. What does it mean in terms of physics? That's quite important. So if we look at a two-level system and have a look at what physically happens when we absorb the pump pulse, we will be able to see what that minus sign means. So initially we have 100% of the population in the ground state, 100% in the ground state. Okay, we then send in the pump. We send it here actually, and the result will be that certain proportion of the population of the ground state will go to the excited state. So we will cut a little bit from the ground state population, and that percentage of the population will appear in the excited state. So if you look at how we measure, that's what will be done by green. When we measure the absorption on the left hand side of this diagram, so we send the probe and look at the absorption, there will be certain absorption because everything comes from the ground state in the absorption. Whereas the probe for the excited system will first see less absorption because there is less than 100% of probability of finding the system in the ground state. And there is also going to be stimulated emission. Stimulated emission. So there will be basically there will be two contributions for which from which we obtain less absorption than in the standard situation for a two-level system. So we first will have absorption, that's the absorption without pump, and the second absorption that we'll do with pump will be less. And as a result, delta A, or in our case the alpha 3 bar, will have an overall minus sign. This will be the pump probe signal. So overall minus sign. There are, in different spectroscopic methods, different conventions about this. For instance, here in pump probe, really, when you have less absorption, so you have the processes of ground state bleach, that's when you have less things in the ground state, and stimulated emission, that's when you have something in the excited state, you have them contributing negative signal. In some other spectroscopies, we will discuss very soon, two-dimensional coherent spectroscopy, the convention is exactly the opposite, but that should not distract you from actually seeing that the methods are clearly based on one and the same theory. The minus sign in the signal comes automatically in our definitions. All right, so there are few important conclusions that we have to make here. One is that pump probe does not have the phase problem. Then that ground state bleach and stimulated emission lead to less absorption, and they have a negative sign, therefore, in the conventions of pump probe spectroscopy. And something that you will see later is that we can use pump probe situation, which has a well-defined phase of the background with which we actually do the so-called homodyne detection. That is, we detect with the inducing field itself. We will use the fact that in pump probe it is well defined to set a phase also for local oscillators that do not work as fields inducing dynamics. In order to calculate the signal that corresponds to the pump probe spectrum, we have to consider all possible Liouville pathways that come in question. And because we know that for a two-level system with which we stay, we only have so far 4 different pathways, it would make sense to look at them and try to interpret their contributions to the spectrum one by one. We need to realize that we have a special case in the pump probe spectroscopy, namely the case that the first interval of the Liouville pathways has a length 0. So we will assume an impulsive limit, that is, the pulses are proportional to delta function, and they are centered at certain specific times and have basically a time width of 0. In that case, the signal directly corresponds to the response functions. Response functions are not integrated with anything, or in fact they are integrated, but the integration leads just to a multiplication of the response function by the amplitude of the pulse. And we have 4 different pathways. I will write them out here and group them into 2 different groups. So we have R1 to R4. I will try to write them out or group them somewhat differently. So the first diagram I will consider is a rephasing diagram in which by 2 interactions we will get to the excited state. Then the next interaction comes here from the right hand side. We are considering positively signed diagrams, and we have E and G, and as a result of the last interaction we would get down to GG. So this contribution gives us non-zero and positive signals, and we recognize that this is the R2 pathway. And an important thing to realize is that there is a tau equal 0 here. With this diagram, I will consider in the same group the other diagram that leads to the population of the excited state, and that's the one which has the order of interactions from left and right exactly the opposite. But clearly, because tau is equal to 0, these 2 diagrams will have exactly the same contribution. So we will have here the interaction which brings us to the ground state. Here we have the excited state, and there is basically an emitted signal here. So these 2 diagrams, if we consider tau equals 0, they actually collapse onto one and the same diagram. This is the diagram R1. So what we have here as an interesting thing is that we have a rephasing and non-rephasing signal. We can actually sum them up because if this is a signal that we detect due to tau equals 0, we do not have any way how to distinguish these 2 signals in the experiment. They go to the same direction and they go through the same set of states. So this is a signal that goes through, or the pathway goes through the state E. So the system is in the middle interval of length t, is in the excited state, E. So with that I want to contrast, but there will be not any real contrast here, but the other group of states will be again certain rephasing pathway, this time R3, and a certain non-rephasing pathway. So rephasing pathway starts with the interaction from the right hand side. So that's gonna be this. Then there is a interaction again from the right hand side, and we end up with G, and an interaction from the left. We go to E and an emission. So here we can see that the middle interval goes through the ground state, and this is the rephasing pathway. And the last pathway, R4, is the one which also goes through the ground state. So we have here on the left hand side pathways that go through the excited state, and on the right hand side the pathways that go through the ground state. And we always have the upper one of the rephasing type and the lower one of the non-rephasing type. So if we actually complete these tasks of writing out all those pathways, we will end up with the following R4 pathway, which has all the interactions from the left hand side. Okay, so this is it. And the important thing is that because we have tau equal 0, it only depends whether the interactions occur both from the same side, the first two interactions, like here, they occur at the same time. So we start with GG and end with GG, or they occur from one and then the other side, or simultaneously from the other side. So from GG we end up with EE. So that's the difference between the pathways on the left hand side and the pathways on the right hand side. Okay, so again here on the right hand side we have rephasing plus non-rephasing pathways or signal, and this one goes, or the pathways go through the state G. Okay, we know that all four of these pathways actually do have a positive sign. So they, well, positive sign meaning that the expression that we derive from the diagrams has an overall positive sign, but we know that this corresponds or contributes to the signal with a negative contribution, that is less absorption. It is how we have concluded that a two-level system will respond, with basically seeing less absorption after the action of the pump. So once we probe the pumped system, there will be less absorption. Now we know that pump probe experiment is done so that the signal that we measure is the one going into the direction minus K1 plus K2 plus K3, with the condition that K1 equals K2. And when the order of interactions is first pulse, second pulse, third pulse, we get rephasing signal. That is the upper two diagrams, R2 and R3 response. And when the order is 2, 1, 3, we get non-rephasing contribution. Now in a real impulsive pump probe, this doesn't matter. Actually, we have to sum up rephasing and non-rephasing contributions anyway. But in fact, we have to do the same in the situation of finite pulses too, because the situation in a finite pulse case is the following. We have the time axis. Here we will have the first pulse, the pulse with K1, and the second pulse is the same pulse, so there is no difference. I mean, this is exactly the same pulse, K2, and actually these two are equal to each other. And now In lecture 6, we have discussed how, in fact, when we have finite pulses, we integrate not over the time tau and capital T, which we normally see in the response function in the impulsive case, but we integrate over the pulse envelopes, basically. And what can happen is that we would have, for instance, two contributions. So the time tau 1 will have to be between two interactions with the same pulse. So we would have tau 1 this way, let's say first being the interaction from the blue pulse here, so from the K1, and then an interaction from the K2. They are the same, doesn't matter, but there will be both the order 1, 2, 3. Let me finish the other contribution. So this is tau 1, there is tau 2, which leads us to the third pulse with the K3, and let's use again the same color to denote where the interaction is, and then here we go with tau 3 to the actual time t where we measure the signal. So this is what happens. We have the order 1, 2, 3, but at the same time, because the pulses K1 and K2 are basically the same pulse, we also have exactly the same amount of 2, 1, 3. So we would have the same amount of rephasing and non-rephasing signals in the pump probe. In fact, we will have to consider always the pairs of rephasing and non-rephasing signal for each of the groups here, from the pathways, for the pathways that go through the excited state and for the pathways that go through the ground state. And of course, we can then actually give an interpretation to these two sets of pathways. So on the left-hand side, we will have the contribution that goes through the excited state. That means it actually depends on how much you excite the system. So this should be interpreted as stimulated emission. So we have stimulated emission contribution. That is going to be the, let's say, the pathways that are involved in that, the response involved with that. Let's call it RSE, and that's going to be the R2 plus R1 pathways. R2 plus R1, whereas the R2 is the rephasing contribution and R1 is the non-rephasing contribution. And as we said, they always have to come in these pairs. We can also write, because of the diagram that we have written, we can write how they will depend on the delay times. So we can see that the last interval, the most upper interval, is the interval in which we propagate the coherence EG. The middle interval is the excited state depending on T, and in the first interval is GE, but tau is equal to 0, so this equals to 1. And we also have a contribution that goes through the ground state. And interestingly, the contribution looks almost the same. So this pretty much can be interpreted as the so-called ground state bleach. That is a signal by which there is less absorption from the ground state. So this is R3 plus R4 contributions. Again, we can see that there is a rephasing and non-rephasing part of this, and they contribute sort of equally. And looking at the composition in terms of the evolution superoperators, the last term is always the same. That's almost per definition, because we can put the last interaction on the left hand side and right hand side equivalently. So we choose the one where we always have u, eg, eg, and then there is u, g, 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 g. That's the ground state evolution, and the last interval here is, well, in case of the GE, this would be the rephasing contribution, but because again tau is equal 0, this gives 1. The R3 and R4, or let's say R2 versus R1, they differ by the first interval. They are rephasing or non-rephasing, but in fact, because tau is equal to 0, the contributions are exactly the same. So there are two things here to note. We cannot distinguish between rephasing and non-rephasing in pump probe experiment, and actually from what we see here, the contributions from the ground state bleach and stimulated emission, they are basically the same. The difference though is, or they are the same only in the situation where the ground state and excited states are both stable and there's no emission from the excited state except for the stimulated emission. So this is how we would approach calculation of pump probe for a two-level system which lives infinitely long. We would see a static signal because, you know, in fact the UEE, EE and UGG, they both in that formulation would be 1, and we would have the stimulated emission and ground state bleach exactly the same and not depending on the time capital T. So the spectrum would basically be for all parameters available always the same. So let us have a look at a situation which is more general, and of course first situation in which we can from which we can see and conclude that pump probe experiment can actually follow some sort of excited state dynamics. We will study the situation in which a two-level system has a finite lifetime. So we have finite lifetime. Of course, both the ground and excited states can have, and actually do have, finite lifetime. However, in a situation which is standard in optical spectroscopy, where the energy gaps are really huge, what is actually significantly constrained by lifetime is only the excited state. So we will have to now extend the way that we describe our experiment by further Liouville pathways that describe processes that we have not considered so far, namely the process of de-excitation of the excited state. So let's have a look how a population of the state E and G evolves in a more general situation of a finite lifetime. So we will first assume that we have still a two-level system, ground and excited state, and that we have a process in which the excited state is depopulated with some rate kGe. The rates usually, at least in my notation, and I recommend this notation very much, has the notation looks so that the state from which you start is the one on the right hand side of these two indices. So it's from E to G that the process goes. Okay, if we have a rate like this, or if we have a general set of rates, we could write kinetic equations for the populations of states. We don't have to do this for two-state system, but for a general one. So if we have a vector of populations, or population probabilities, that contains some number of states, the equations of motion for this system often can be written in a way that it's a differential equation in time, where this vector of the population elements would be multiplied by a matrix of rate constants or rates. One thing is that they don't have to be constant. So in fact we can have rates that would look like this, and here you can see why it is important to keep the order of indices in the way that I suggested, because in these equations this all works like a matrix multiplication. So for instance, for the rate of change of the P1, we would have contributions K11T, P1T plus K12, P2, etc. So the contributions from 2 to 1 will be driven by the rate K12, which can be in principle time dependent. The rate K11 will have to be negative. So this is minus. In fact, it has to be minus sum over all other states, depopulations of the K1 to the other states. So this would be a sum over n from 1. But that doesn't matter. That's a detail. The important thing is that once you solve these equations, and because you know quantum mechanics, this is not going to be a surprise to you. If you imagine that instead of p's you would have elements of a state vector, you know that you can write an evolution operator. So the solution here could also be written in terms of some generalized evolution operator. So P1, P2, etc. This vector could be written, and now there is no time derivative of it. This could be written in terms of a matrix of time evolutions. Which will be applied to the initial condition, so P10, P20, etc. So that would be the solution, and these would be the use here would be elements of some sort of an evolution operator which acts on not on a quantum mechanical state vector, but on a vector of populations. And you can see that P1T could be written as U11T P1T plus U1T P2, sorry, this should be here 0 and 0, etc. So here are the initial conditions, and what is then actually the U11 and U12? In fact, these things have a very straightforward interpretation, which will be very good and very reasonable for our further interpretation of the components of the response functions. Namely, the U11 T is in fact a conditional probability. We will explain. Probability that the system will start in state 1 and after time t also end in state 1. So imagine that that the probability of being in state P1 is 1, then actually U11T exactly corresponds to P1. So it's a probability. It's the probability of being in 1, and that's only in the case that P10 is 1. And if P10 is 0, so there still can be P1, but U11 cannot contribute, or P10 therefore cannot contribute. So the U11s clearly as the function of probability of being found in state 1, but with the condition that you were at the state 1 at the beginning. Yeah? And very similarly, U1, 2 is a conditional probability that the system will start in state 2 and after time t ends up in state 1. Again, basically exactly the same situation. If P2 0 were 1, that means also that P1 0 is 0 and so on. Then P1 equals precisely U1 2. The probability of being found in the state 1 is exactly U1 2t if P2 is equal 1. And of course, with P2 being less than 1, the probability or contribution of that part to the probability will decrease. So it's a conditional probability. That's a very important conclusion here. Okay, now we actually do already have such elements present. So this is, if you remember, in fact, the Pn T here corresponds to the density matrix element rho n T, and therefore if you have P n T equal U n M T P M T, and of course all the other contributions, this corresponds in the language of this 2 indexed row, this corresponds to a contribution U and N M M T rho M M T plus the other contributions. Yeah. So it's clear that instead of the use with 2 indices, we can use these calligraphic use with 4 indices, and the interpretation of the terms that we already have in our response functions is the following: U E E E E T is a conditional probability, and now I don't have to actually write the rest, that you start in E and end up again in E. Yeah, the same thing, same interpretation for GG. GG, it's again conditional probability and here we have the GG. But obviously, because we have we can assume that there is a transfer from E to G, we must have also a contribution which starts in the E and ends with G, and again this goes from the right hand side to the left hand side. So this is now the conditional probability that the system starts in state E and ends up in state G. That is a process that definitely occurs. Well, we can ask ourselves, how is this process looking like, and how the corresponding element of density matrix would look like. For instance, if there is a process of transfer from excited state to the ground state, This also has to influence the UEE. So that's the first thing that we will consider. Obviously, if there is no process from the ground state to the excited state, the UGGGG still is actually fixed. So, well, we start considering the relaxation. So we can write that the population of the excited state is, if this is the only process, there is a process of de-exciting with the rate GE, and that's the only thing that we have. This has a very simple solution. This is PET equals e to the power of minus k GE T PE 0, and then we clearly can conclude that this is actually the UEEEE. So we can write UEEEET equals to e to the power of minus k GET. So we found one contribution now in this decaying case, which is already different from the case where there was no energy transfer. So the pathways that go through the excited state now have to have this factor e to the power of minus k GET. Yeah. Now we also know that this one keeps to be the same, but that does not mean that the population of the ground state does not change. It does, but because this is a conditional probability of the process in which you start with G and end up with G, this still is 1, because nothing gets from from ground state to the excited state. Now we can actually find out relatively easily what is the UGGEET. This will come with a question mark, but we can fix it relatively easily. Look at the following. We have a condition. We have a condition that PET plus PGT are equal 1, and namely for all times. So at the beginning, but also at the very long times. Now we also know how PE looks like. So we can write that this is e to the power of minus k GET PE at 0 plus, we can now write the PG, but the PG will have a contribution that originates from U. Okay, we can maybe write this somewhere else. So for instance, we can write it here. PGT will be UGGT PG0 plus UGGEE PE0, and this is thing that we have to enter into our equations now. So we will put this in. We know that UGGGG is 1, so we can write that this is PG0 plus our unknown UGGEET PE0, and that this is supposed to be equal 1. And now we can write PG0 in terms of the PE0. So this is e to the power of minus kGET PE0 plus, and here we will have 1 minus PG0, no, PE0 actually, PE0 plus UGGEET PE0, and that equals 1. We see also that the 1 on the left hand side and right hand side can be cancelled, and we can isolate we can actually then also divide everything by PE equals 0, and the result to which we come is relatively easy to understand. We get EGGEET PE0 equals 1 minus e to the power of minus KEGT PE0. So this for all PE0s means that UGGEET equals 1 minus e to the power of KET, and that's an important result that we can use. Well, I think that this is of course enough for the way that we treat the problems now, but there is also, it makes sense to discuss this particular process of establishing the UGGEE in somewhat more detail. We might actually work through a more general expression to UGGEET than what we have now. So more general expression for UGGEET might come handy in interpreting what's actually going on. Well, we can still keep working with some conditional probabilities. For instance, the conditional probability to jump from E to G in unit time is the relaxation rate. So the conditional probability to jump from E to G from these states in unit time equals KGE. That's basically the definition of the rate constant here. Give it some time to think about this, why that is. Imagine that you have the equation that looks like this. And imagine that, for instance, PE is always 1. Now, when you integrate this, you get that PG, the probability, equals minus KGET. So KGET is a probability. So KGE is a probability in unit time. Well, of course, that would be a very strange equation if PG would grow and PE not, but this was only to illustrate how is this KGE to be interpreted. In fact, there is a correct way to interpret it. That's what I will write now. One that doesn't look so crazy as here, and that is when you consider only processes that happen on infinitesimal time scale. So if you ask what's the conditional probability to jump from E to G in time interval t from t to t plus dt, then that is actually KGE dt. That's a correct interpretation because you can assume that on the infinitesimal time scales the probabilities do not change. So we can, for instance, ask ourselves what is this conditional contribution to the population of the ground state that comes from the excited state. So we will ask ourselves what is the PGT, but on the condition that it came from the excited state. And that has to be all the jumps at all times t. So we will integrate from 0 to t d tau over all the possible times jumps. And that's this actually. At every time we will have d tau KGE probability, conditional probability that we jump. So if we multiply it with PE tau, we have a probability that we have jumped at that time tau. And now we integrate from time from 0 to t. So what we get here is integral from 0 to t d tau k g e u e e e e tau PE0. We wrote PE tau in terms of the element UEE EE. And now, because here on what we have here on the left hand side, that's actually UGGEET by definition, PE0. We can see that that is equal to the integral that we wrote here on the right hand side, PE0. So, because PE0 is immaterial here, we can contribute that UGGEET is equal integral over T over tau. KGE will be also present here, and UEE tau. Now that means, because we know what is UEE, that means that that is KGE integral e to the power of minus KGE tau, and that gives us 1 minus e to the power of minus KGE T. So that's exactly the result that we've obtained above, but this is actually the more general expression here. We integrate over the depopulating element of the evolution superoperator. Okay, so now the only thing to do would be actually to put all these results into the response functions and devise contribution to the response functions that also take into account the contribution for the transfer of the state from E to G. So in the next video we will have to extend the set of Liouville pathways by which we described a two-level system, and this will give us some sort of a blueprint for further extension of the set of Liouville pathways to systems of more than 2 levels, both actually adding states in the excited state band and in fact adding yet another band of excited states that will be above the standard band that we discuss now, and that will contribute to the process of so-called excited state absorption.